Hi, this is Miss Litton, and this is AP Biology, Chapter 9, Review, Cell Cycle and Reproduction, all about mitosis. Say hi.
Hi.
We have a large group here, heavily motivated. Okay, first thing, when I show you this, not really Miss Litton, when I look at this, could you understand everything that's on this introductory diagram that we have talked about?
Yes.
Okay, so we talked about the cell cycle in general, multiple times, right? And then we talked about the controls with it. We talked about the details of mitosis and what happens at each stage. And then we said what happens when we lose those checkpoints and cancer. That was all this chapter, right? I'm going to update that quiz. Okay, so let's hit all the big ticket items. First thing is knowing the cell cycle. That's easy peasy lemon squeezy. Don't eat food that is too greasy. Right. So I expect you, when I ask you about the cell cycle, remember how we talked about it in class and I had you diagram it in class? I expect you to feel comfortable with doing that, okay? And so just quickly going over the big ticket items, we could start anywhere because it's a cycle, but let's start with G1. What do we know about what happens during G1?
Organelles.
Here, I'm going to move there. Yes, organelles, more organelles. Why does that logically make sense to us so we don't need to bother memorizing that? Yes?
The cell's just been split.
Cells just divided so it's half the size. Makes sense we need more organelles. Yes? We also have a checkpoint there. Why does that make sense to us? We just split. We want to make sure it's okay. In fact, we know, we learned about, as far as control of cancer, right? Cancer. Remember the two categories of genes? Don't shout it, just think it in your head. Don't whisper it. Don't whisper it. Just think it in your head. Does one of them start with a P? And one of them starts with a T? Okay, tell me the P one. What was that word? Proto-Oncogenes, right?
Product. I like product.
Okay, Proto-Oncogenes. What was the other one? Tumor suppressor gene. All right. So let's just review that just for uno momento. Proto-Oncogenes are genes that are in a position of power and control. So if they go bad, a lot of things go bad, right? Proto-Oncogenes are ones that facilitate the cell cycle. Say go, go, go. They promote the cell cycle and prevent what? Apoptosis. The other category of genes, not proto-oncogenes, what's the other category? What are they? Tumor suppressor genes. Tumor suppressor genes say cell cycle what? Stop. Okay, and the second thing they'll do if they can't fix it is what? Kill it. Do you remember a key one of the tumor suppressor genes was P53? Remember that? P53 plays a, oh, it's right there. P53 gene functional. See it in the diagram? I was leading to that, but it was already on the diagram. Okay, that's where P53 plays a big role. P53 would be your example of a tumor suppressor gene. A proto-oncogene, which goes bad, it's an oncogene, right? Position of control. How do we remember that one? It's like stepping on the gas. What's its name? RAS. Okay, RAS triggers the cell cycle. So that's your example, right? So there's two gene categories and then an example for each. Two gene categories related to loss of control that we could have and have cancer. One is proto-oncogenes, positions of control could go bad. Our example of that is RAS. And it's not supposed to trigger the promotion of the cell cycle unless triggered by like a receptor, like a growth hormone triggering it. But it just goes on its own. It's a bad RAS. Second category is tumor suppressor genes. An example of a tumor suppressor gene we want to know is P53. Okay, you tying it back into this point? Okay. Another thing that has to happen before we go forward into S, a form of control, is we have to increase the number of what? Cyclins, right? We have to have a ticket and we have to go through security. So that was the security, but we also have to have a ticket. And in this case, we need S cyclin. And we need S cyclin to bind to who? CDK. What does that stand for? Cyclin-dependent kinases, right? So those cyclin-dependent kinases, ASE, ACE tells me it's a enzyme that's going to facilitate the next step. What's the next step? Synthesis of DNA. This is where DNA replicates. In our mind, if we had a cell that had 10 chromosomes, we got that? 10 chromosomes total. What would you say that is? It's N number or 2N? I would say, hey, this cell has 10 chromosomes in it. 2N, right? Okay, how many homologous pairs does it have? 5 homologous pairs, because 5 times 2 is 10. So it has 5 homologous pairs. That would be its what number? N number, right? Haploid number, exactly. Okay? So if I have 10 chromosomes in my cell, normal cell, I'm doing my arms, my legs, my mitosis. I'm going to do DNA synthesis. For a while there, each chromosome is now going to consist, after the S stage, each chromosome is going to consist of what? 2 sister chromatids. Does that make sense? I've doubled the DNA at this point, after S. Why do I need double the DNA? Because I'm heading towards cell division. And I'm going to give a copy to each daughter cell. We all right with that? Okay. So once I've gone through the S stage, I'm ready to move into G2. But again, I'm going to have a what first? Checkpoint to make sure that I did DNA replication correctly. Okay, now I have a cyclin that's building up. What cyclin's going to build up? M cyclin. Why? Getting ready for mitosis. Okay, now I'm ready to enter the stages of mitosis. And when I think about the stages of mitosis, which I'm thinking about right now, here they are, I'm thinking about them. When I'm thinking about the stages of mitosis, I'm thinking the first stage is called what? Prophase. And I know the chromosomes, do it with me. The chromosomes become visible, but when they become visible as what? Sister chromatids. They were always pairs, right? Homologous pairs, but they have sister chromatids. Chromosomes become visible, the nuclear envelope and the nucleolus disappears, it breaks down. Radiating microtubules, asters, they meet in the middle at metaphase. Who meets? Do they meet side by side? No, that's for what? Meiosis, when you're getting ready for sex. We're just doing growth and repair, okay? Or asexual reproduction, so we're going to line them all up. They meet in the middle at metaphase, away at anaphase, 2 nuclei at telophase, cytokinesis. If I pinch off, what's that called? Cleavage furrow. Who does cleavage furrow? Animal cells, because they don't have a cell wall. If I'm a plant cell, I got to build a cell plate. Yeah, first a cell plate, and then it becomes a more compact cell wall. And that's how I divide into 2 cells. Okay, so I can see prophase, metaphase, yeah, we did all of that. Beautiful. Okay, so our body cells are called somatic cells, and they do mitosis. Okay? The only thing that does meiosis, right, is when we're forming gametes, either in our ovaries or our testes. Yeah, we keep them up here. Okay, ovaries or our testes. Okay, but plant cells, their apical meristems at the tops of their shoots and the bottoms of their roots are always continuing to grow. And if you're a eukaryotic, you have secondary growth from your vascular cambium. But I don't really think we put that in the notes yet. I just said that part, right? Okay. All right. Let's check for understanding. See if you will ace this. Today we are going in as our favorite spice. Seasoning or spice.
I know all about spices.
My favorite spice.
We know all about spices.
Your favorite spice. I'm going to pause for a minute. All right, let's check. Yay! 50. Wait, whoa. Everybody got it. Oh, yay! People. That's fantabulous. Oh, losers. Okay. Come back to me so we can address them. They said proteins are being synthesized in interphase S. Could someone want proteins to be synthesized? Sure. But what's S all about? Your DNA replication. Oh, one person. Sister chromatids are separating away at anaphase, not away at telophase. Oh. Division of the cytoplasm, cytokinesis. Membrane breaks down, prophase. Oh, so, okay. Organelles are formed, G1, DNA, good. Everybody got it that got to it. Okay. Uh-oh. Reforming. Nuclear envelope reforming is telophase. Tell your bio buddy, how'd you do on that? All right. Come back. Come back. You're too yappy. You're too yappy. Good cells going bad. You know, oh, side note to get you ready for your essays. Okay? Mitosis, I could say two different, actually mitosis and meiosis. I could say, I have a cell whose 2N number is 6. Draw me metaphase of mitosis. Conversely, draw me metaphase 1 of meiosis. Draw me metaphase 2 of meiosis. Now, we've just started in on meiosis, but I'm going to tell you, that's the kind of question I might ask. Because I know if you know it, if you can draw all those stages. Does that make sense? What would the chromosomes look like in straight up metaphase? What would they look in meiosis 1? What would they look like in meiosis 2? That's the kind of essay question. Okay? When we finish chapter 10 tomorrow, ask me to revisit that. Okay? When we finish chapter 10 tomorrow, ask me to revisit that, and we can try that and compare and contrast what it would look like in all those stages. Yes? I can't hear. Shh, go.
In an essay question, could you ask us to diagram the cell cycle of mitosis?
I could totally ask that. Now, it's not the kind of question you'd get on the AP exam, but I can know real quick if you understand things and have all this, everything set in your head. That's why I would do that. I could ask you a question about what are the controls of mitosis and why are they important. I could ask you to rank the controls, which ones are most important. So I can have you ascribe value, right, to them so you can tell me what's most important. You remember that question I asked you on your Unit 1 exam where I said, pick the 4 most important organelles, and some of you didn't even pick nucleus? Yeah. Okay.
Unless nucleus was excluded.
Yeah, unless nucleus was excluded, and some of you had that. Okay. Another kind of question I could ask is, what are the characteristics of a cancer cell? How would you know a cancer cell is different from a normal cell by appearances? If you were looking at one under a microscope, could you come up with some things? Because remember when you were role-playing back and forth? I'm cancer, I'm not cancer, right? You know, what kind of controls ensure that you won't have a cancer cell? Those are the kinds of things I might ask you about, okay? So, coming back here, we talked about this is something that cells have in place that would prevent cancer, right? Uncontrollable growth. These are normal things that cells have. They have density-dependent inhibition. When they're crowded, they stop dividing. It's a normal thing that cells would have. Most cells have what's called anchorage dependence, which means they have to be attached to some substratum, some base, some other cell layer, or some membrane. And if they're not, they won't divide. Obviously, exceptions, things like red blood cells and white blood cells, right? But those are things put in place already that prevent you from being a cancerous cell. Because if you become detached and you're starting to move through the body, you won't divide because you can't divide unless you're attached to something, right? Unless you have some sort of mutation and you're a cancer cell. So that's when we talked about that you have a series of mutations before you become cancerous. You have to lose contact inhibition. You have to lose anchorage dependence. Just because you lose those two things doesn't mean you're going to have cancer. No, but if you've lost those two things and then you lose control of the cell cycle, then you could, right? Because then those cells could spread to other places. Do you see the big picture there? Okay. Now, cancer cells, like we said here, okay, is this one. It looks like it still wants to adhere to things, so that part it hasn't lost. but it has lost contact what? Inhibition. Okay? Now, that grouping of cells could end up just being a benign tumor if it does not escape where it is. If it's encapsulated in some sort of membrane. If it just stays there and goes nowhere, you go in and you cut it out and you're all done with it, right? The concern is when it metastasizes and some cells can leave its cancer home base and spread to other places in your body, right? That's when you're concerned, when it metastasizes. Now, that process of metastasizing, we looked at, obviously, just even have cancer, you're not following the cell cycle, but B, you can spread to other places. We talked about carcinogenesis is a series of mutations, usually. So one thing after another has to go wrong before you would be considered cancer. This was a good diagram that explains how do normal cells operate, how do cancerous cells operate. So normal cells have a specific structure, they have probably structure to function, but these are non-differentiated. They have normal nuclei. You know what a nucleus looks like, but these have abnormal nuclei. And specifically, if we go to nuclei, we can see here they have multiple nuclei, they're large and really coarse chromatin. So the DNA looks different. Okay, other things is they don't undergo apoptosis, right? They're the immortal cells. Maybe they have a little telomerase going on, making their telomeres longer, and they keep dividing and dividing. They exhibit no contact inhibition. We know that. They keep crowding out. They're disorganized, multi-layered, and they're spreading. Angiogenesis. What was angiogenesis? Do you remember that? Yeah, blood vessels grow towards that cancer in order to supply it with nutrients. And the bad thing there is you could have a tumor, okay, that's there, but it hasn't been able to spread because it doesn't have any access to any other places. And it doesn't have the nutrients. So then you have angiogenesis. You want to get rid of that tumor before angiogenesis takes place, supplies it with nutrients, and then gives it a vehicle to get away and go to other places in your body. That's why it's so important to catch those things earlier. Okay, then this brings us to what we discussed already at the very beginning, these controls. We have two different groups of genes. And remember, we talked about proto-oncogenes. There's over 100 different proto-oncogenes that could go bad. You know this because remember the complicated cell cycle that we looked at, and there are all the different things in there? So you could make one of those products that could throw off the whole cell cycle because there's so many things involved in pushing it forward. So those proto-oncogenes are not bad. They're just in a position of power. And if they go bad, they're going to promote the cell cycle and work to prevent apoptosis. Your second category, who was our example of a proto-oncogene? He's a general, yes, he's General Petraeus, right? But who is the real one in your body? What one is that one? RAS. Remember that? RAS is like stepping on the gas, okay? And it's saying go cell cycle, go, and let's not kill ourselves, okay? The tumor suppressor genes, these genes are coding to slow down the cell cycle so we can check to make sure everything's all right and go through a checkpoint. And if we can't fix it at the checkpoint, let's just kill ourselves, okay? And who was your example? RAS is our example here. Who is our example here? P53, okay, was our example here. And here was just your summary on proto-oncogenes. Okay, and then we talked about that there were 100 different ones, and that we talked about how it would promote the cell cycle even if it hadn't received a signal to move forward. It went ahead and moved forward. And we looked at RAS, who, if it's mutated, it'll promote the cell cycle even though it has not been triggered. We look at the breast cancer genes, and we realized that those are on autosomes, and males and females could pass those on. We then looked at the tumor suppressor genes, and we said you need to stop and check to make sure everything's okay and fix it, and if you can't fix it, then you kill it. And we specifically looked at how P53 is involved, and we, put this back into what we had already learned about CDKs, the enzymes you need to be functional in order to promote the cell cycle. And it will make proteins, code for proteins, that will bind to CDK. So who can't bind to the CDK? Cyclins. If cyclins can't bind to the CDK, you can't move forward in the cell cycle, right? Because you have a product from having CDK and the cyclins bound together. So that causes it to stop. That's the slowing process. And then here, caspase, remember, Casper, the friendly ghost you, kill you. Sounds, yeah. So we said failure of DNA systems can cause that, like we have corrective measures, so if you don't correct it, it gets through. And then we talked about telomerase, because every single time a cell divides, it loses a little bit off the end of its chromosome, and that brings it to its natural death of a cell. It's going into G0 and it's done. But when you have telomerase, it keeps the ends long, and that keeps it that immortal, that immortality, because it keeps allowing it to keep dividing and keep dividing and keep dividing. Okay, then we just talked about different tests we could do, right? Blood tests. We were real relieved to find out about the prostate. Okay, and then we went into yes?
For the cyclins of the CDKs you're talking about, are they MPFs? Are they types of CDKs?
Okay, so let's go back here. It's a product of the CDK with the cyclin.
Okay, and you need, and CDKs bond to S and M cyclin?
Yes.
Okay.
S cyclins when you're going into the S stage, and M cyclins when you're going into the M stage. Does that help you?
Yes.
Okay. So then, okay. So if we're going to talk about, we know there's life, right? Then there's two categories of cells in all life. What are the two categories? Prokaryotic and eukaryotic. Eukaryotic cells, right, have linear chromosomes, and they do mitosis for growth and repair and asexual reproduction, or they do what? Meiosis to get ready for sex. Prokaryotic cells, what two domains are under the umbrella of bacteria and archaea? They have circular chromosomes, both of them. And the way they reproduce is through binary fission. So the DNA replicates, it attaches to two sides, and it's kind of like doing a cleavage furrow, but not really, because it has a cell wall. Okay, so that's how they operate. And I think that was it on that. Awesome blossom, yes.
Yeah, so
Wait, stop, I gotta hear the question. Go.
just checking, are a functioning proto-oncogene, and is it in charge of committing apoptosis?
Okay, so let's talk about that. Is a functioning proto-oncogene in charge of committing apoptosis? Remember, proto-oncogenes are saying yes to the cell cycle normally, and they're preventing what? Apoptosis. Because they're about, let's make more cells because we need them.
Perfect. So the tumor suppressors are the ones that actually are in charge of initiating the
Yes.
apoptosis. Okay.
Yes, exactly, yes.
So, you say that like the proto-oncogenes, they prevent apoptosis even when they're considered good?
Yeah, what they're about is, let's say you need a bunch of killer T or immune system cells to fight some sickness that you have, right? You don't want those cells to die. Right now, you want to make a bunch of those B or T cells to help you fight that infection. You're going to do a bunch of that cell cycle, make all those cells, and prevent apoptosis because you want them to live. Once the infection has been completed, you do want them to commit suicide. And so then there'll be a trigger for that. It's not so much that they're about preventing apoptosis in the sense like, wow, that's bad because those cells have to die. At that point, you're trying to make more cells, so you're not about the death stage. You're about, let's make some cells. Let's make them and not kill the ones we're making. Well, it's just the opposite of tumor suppressor. And remember, both have to go bad in order for you to actually have cancer. Guys, are you okay? Because you're really distracting me. Okay? Any other questions on that? Okay, let's riddle me this, spicy children. We went as spice if you came in later. Squid ink is your spice?
Spicy. I knew it. We know too.
Do we really like squid ink?
This is a joke.
Oh, I'm going to pause you. See how you did. Yay, 51 of you got that right. Oh, When cancer occurs, it could be a result of mutations or the RAS genes being operational without any external signal, meaning you're not getting a growth factor. Not due to these. 3. Which of the following is not a characteristic of cancer cells? They are not undergoing apoptosis. That's the problem. They should and they're not. Number 4, if the cell is cancerous, you might find an abnormality in all of those things, a receptor, a signaling protein, and a gene. And, children. Okay, make good choices, which involves no smoking. Stop and listen. If you're going to return the test to me, period 1, period 3, period 5, we won't put them in order because there's just too many right now. You may look at them. You may ask me questions up for the next 2 minutes. Have a piece of toast. Make good choices.